Some of you may have seen a similar talk that I gave at the last Padua muscle meeting. So some of this may be familiar to you, but what I'm going to talk about today is what is mitochondrial permeability transition, which I will abbreviate to MPT. And I'll allude a little bit to how it's affected with aging, because that is our primary area of interest, but I'm really just going to focus on the event today and some of its consequences. So the way we got into this is we've had a long-standing interest in the mechanisms that drive skeletal muscle atrophy and contractile dysfunction along the continuum of the life course. And mitochondria have been implicated in this process for many years by a wide number of different groups. But my reading of the literature to this point is that our understanding of the precise mechanisms by which the mitochondria may be involved, it's still a bit unclear how the specifics are involved here. And one of the particular areas that I think warrants additional research is this event, mitochondrial permeability transition, which many of you will know is regulated by the mitochondrial permeability transition pore, or MPTP. So the structural components of this pore are still pretty heavily researched. I think I saw Dr. Paolo Bernardi on the line. He, of course, has contributed a lot of research to this area. And while there's still some work to be done, I think that the horses are kind of marshaling around a couple of different components. So one being the ATP synthase as a major contributor to the pore. For example, in artificial membrane systems, either monomers or dimers of the ATP synthase exhibit pore-like activities. And then another one that seems to be particularly important is the adenine nucleotide transporter, or ADP-ATP carrier. So whether these things are separate pores that have different conductances, that's certainly been suggested, or whether they operate in conjunction with one another, I think those are still points to be understood. So there's still work to be done here. But the idea that the permeability transition pore and MPT might be related to aging, there's a fair amount of literature related to this, particularly in the neurodegeneration field. So in neurodegenerative diseases like Alzheimer's and the like, there's pretty good evidence that mitochondria are more sensitized to permeability transition and contribute to the loss of neurons in those brain structures. But in skeletal muscle, the implications of this are far less understood and less studied. So let me give you a bit of background on mitochondrial permeability transition as an event. Many of you will understand that MPT has kind of a continuum of operation. So on a moment-to-moment basis, there are kind of these short transient flickering-like activities that appear to be really important for cellular signaling. Things such as calcium and ROS signaling in the cell are regulated by these transient flickering behavior of the MPT. But what I'm really interested in is kind of this more prolonged mitochondrial permeability transition pore opening because it has more of a consequence in the cell. So I'm just going to go through this for you. What I've got is a little zoom here of a portion of the mitochondrion showing the outer mitochondrial membrane, the intermembrane space, the inner mitochondrial membrane, and the mitochondrial matrix, and a couple of different calcium transporters, the voltage dependent ion channel in the outer membrane, and the mitochondrial calcium uniporter on the inner membrane. So the best understood trigger for mitochondrial permeability transition is calcium. And so when calcium reaches high enough levels in the cell, the mitochondria are, of course, an important buffer to that calcium. The mitochondria take up that calcium. It enters the intermembrane space and then goes to the matrix through the mitochondrial calcium uniporter. If it accumulates to high enough levels, that can trigger formation of the mitochondrial permeability transition pore. And that leads to the release of ROS from the mitochondrial matrix and dissipation of that mitochondrial proton motive force. That can cause swelling of the mitochondrion, which then releases a multiple of mitochondrial constituents, including reactive oxygen species and cytochrome C. And I've highlighted these two because I think that their role in skeletal muscle might be quite important, and I'll elaborate on that a little bit more momentarily. So what evidence do we have that MPT is relevant in aging skeletal muscle? So this is just some data from my lab. The data from men is published, the data from women is not yet published. But what we do is we take a small muscle bundle from elderly subjects obtained by...
Sorry, I'm having trouble hearing you.
Oh, sorry, that's my Siri talking to me. I don't want to talk to her either right now. So we take a small muscle bundle and we saponin permeabilize it and then deplete it of myosin. And then we expose it to high calcium with a calcium green indicator. And we can measure that calcium green fluorescence fluorometrically. And so we're able to determine the time and the kinetics of the calcium uptake before that permeability transition pore event. And what you can see is that both in men and women with aging, there's a shorter time to mitochondrial permeability transition. So the mitochondria are more likely to undergo permeability transition in response to a calcium stress. Now, if we look within a muscle cross-section, that's the image you see in the middle here. We've got dystrophin in green, apoptosis-inducing factor, which is a mitochondrial protein, in red, and then we got DAPI in blue for the nuclei. And what you can see is I've got a zoom in on an inset here to show you a single myonucleus. So you can tell it's a myonucleus because it's under the dystrophin border. And you can see that this myonucleus has got red in it. So that's the apoptosis-inducing factor. Now, the only real way for that to get there is if the mitochondria have undergone permeability transition, had the pore open, and this protein be released into the cytoplasm. So this is evidence that not only are the mitochondria sensitized to undergo permeability transition with aging, but they have also undergone permeability transition. So if we quantify the abundance of these AIF positive nuclei, we find a dramatic increase in aging skeletal muscle. So all of this has kind of led us to question, what is the role of mitochondrial permeability transition in skeletal muscle? Obviously, we're really interested in that in aging, but we're also interested in skeletal muscle in general. And I alluded to earlier that we're particularly interested in a couple of aspects here: mitochondrial ROS release during permeability transition, as well as release of cytochrome C. And the justification for the cytochrome C side is that this will lead to activation of caspase 3. And while caspase 3 is perhaps best known in mononucleated cells as you know, terminal caspase prior to the full induction of cell death through apoptosis, in skeletal muscle, caspase 3 has proteolytic activity and affects a number of different proteins. For example, it affects some of the proteasome regulating subunits to cause an increase in proteasome activation. It also causes cleavage of the actin and myosin from the cross bridges. And it has also been implicated in regulation of a Wnt protein called disheveled, which is essential for the maintenance of the acetylcholine receptor cluster at the neuromuscular junction. So for all of those reasons, we think that the activation of caspase 3 and the increase in mitochondrial ROS that would occur as a result of mitochondrial permeability transition are probably particularly important for skeletal muscle. So here's our mitochondrion undergone permeability transition, and perhaps we've lost a myonucleus. What's the consequence of that? So we are focusing first on whether it might be a mechanism causing muscle atrophy. So to really address this experimentally, the first thing that we did was to develop some chemical strategies for inducing mitochondrial permeability transition. As you saw from Dr. Smuter's talk a few minutes ago, she showed you that doxorubin, that this chemotherapeutic agent, amongst its actions is an increase in reactive oxygen species generation. And that, along with the calcium dyshomeostasis that occurs with doxorubicin, those are both potent triggers for mitochondrial permeability transition. And so what you see here is with doxorubicin treatment, this is in cardiac cells actually, that you get a reduction in this calcium retention capacity, which is a sensitization to mitochondrial permeability transition, and showing that this is related to MPT. If you treat with the MPT inhibiting compound called cyclosporin A, you can prevent this DOX-induced reduction in calcium retention capacity. So you'll see in many of the experiments that I'll talk about momentarily, we've used doxorubicin to induce MPT. But then I'd also like to introduce another compound that I'd like to thank Paulo Bernardi for introducing us to, and it's this benzodiazepine called BZ423. And his group showed back in 2013 that this agent will also reduce the calcium retention capacity of the mitochondria. And other groups have now been using this agent as well. And so I'll show you some data today that's actually hot off the press. My PhD student, Sarah Burke, just gave me the data yesterday. And you'll see how we're using that agent now as well. Okay, so thanks to my collaborator, Terrence Ryan, and Trace Tome here at University of Florida, we've been doing a lot of work with cell culture. And so on the left here, I'm just showing you, this is the time to mitochondrial permeability transition when we use our saponin permeabilized and myosin depleted muscle fiber bundles, incubated with doxorubicin for about 4 hours during the permeabilization and depletion process. And what you see is there's a sensitization or a reduced time to MPT in response to the calcium challenge. And when we look at C2C12 cells that have been treated with doxorubicin, we find that there's an increase in the amount of apoptosis inducing factor, which is released during MPT, associated with nuclei in the C2C12 myotubes. And that is prevented by either inhibiting mitochondrial permeability transition using cyclosporin A, which I've already mentioned to you, or buncreatic acid, which is an inhibitor of the adenine nucleotide transporter, which I mentioned is also one of the proposed constituents of the MPTP. Or you can block mitochondrial ROS with a mitochondrial targeted antioxidant called mitotempo. Each of these things will attenuate the DOX-induced increase in AIF nuclear colocalization. Secondly, if we look at the mitochondrial ROS signal using an agent called MitoSox and look at the area of the MitoSox signal in response to doxorubicin treatment, you can see that DOX dramatically increases that. And again, we can attenuate that with MPT inhibitors. So moving on to look at the atrophy behavior. So this is again, collaboration with Terrence Ryan's group, as well as now Karen Esser's group. So with Terrence's group, we looked at two-dimensional, kind of more standard C2, C12 myotubes. And then with Karen Esser's group, we looked at C2, C12s that had been put in these molds that have basically allow the C2, C12s to grow in more of a three-dimensional, more physiological-like configuration, and they mature a little bit better than do the two-dimensional type. And regardless of which prep we're using with these myotubes, we find that doxorubicin is inducing myotube atrophy. This is a 24-hour incubation. And then again, the MPT inhibitors will prevent that atrophy, and we see that in both the 2D and 3D C2, C12 preps. We've since moved on with my PhD student, Sarah Burke, to look at this in single muscle fibers. These are mouse flexor digitorum brevis muscle fibers from adult mice, where we use a collagenase, and I think there's a couple other agents in there as well to help us separate out the single muscle fibers. And then we can then incubate them in the presence of, in this case, doxorubicin on the left. That will induce atrophy. That can be prevented with cyclosporin A. And then similarly, if we use this benzodiazepine BZ423 that I've mentioned, induces permeability transition, we find that that also induces atrophy. So that's in the white bars here from day 0 to day 1. And that atrophy can be prevented by a new MPT inhibitor called TRU2. Well, we call it TRU2, TR002. It's an analog of an asoxazole agent 63 that is believed to cause direct blocking of the mitochondrial permeability transition pore. And so in both of these settings, we find that inducing permeability transition with a chemical agonist causes atrophy that can be prevented by an MPT inhibitor. We've also looked at this in human primary cells, and it's the same story. We cause atrophy when we treat with chemical inducer of MPT, and again, that's prevented by MPT inhibitors, and we've now expanded the use of MPT inhibitors operating through a number of different mechanisms, cyclosporin A, and buncreatic acid, which I've shown you already, and other cell preps. And then morin, which is a phytochemical found in plants, which has pleiotropic actions, but amongst them is inhibiting MPT. And then that isoxazole 63 that I mentioned, the TR002 being a related chemical to that, that's more stable. And so we find that any of these mechanisms of inhibiting MPT will prevent the DOX-induced atrophy. And then lastly, we are also working with a single fiber model of what we consider to be a disuse/denervation atrophy. So of course, once you isolate these muscle fibers, they're no longer contracting, and they're also no longer innervated. And if we maintain them in culture without any chemical agonists of MPT, so this is just a normal incubation, cell culture incubation media, we can maintain these fibers for up to 7 days. And over a 5-day period, we get almost a 20% atrophy. And just as we're finding with the chemical inducers, we can largely prevent that with an inhibitor of MPT. So under a variety of settings now, both chemical as well as disuse atrophy-like conditions that are associated with mitochondrial permeability transition, we can prevent atrophy by blocking MPT. So how is this working? What are the mechanisms? So I'm just going to finish with a couple of quick slides here. And so one is if we use shRNA targeting caspase-3, we can also largely prevent the DOX-induced atrophy. So it looks like caspase-3 is an important mediator of the MPT-induced atrophy.
3 minutes left.
And then lastly, if we look at some of the signals that are downstream of ROS that have been linked to muscle atrophy, we find that in the context of MPT, that many of these same things are also affected. So if we look at FOXO, we find that it's upregulated with DOX treatment, and that's attenuated with boncrucic acid, which is an inhibitor of MPT. Same idea with the ubiquitin ligase MAFBOX, and P62 is part of the autophagy pathway. So it looks like MPT is inducing atrophy through kind of the classical atrophy signals, and our inhibitors are preventing that. So this is our working model, just to finish, that with aging, we're seeing sensitization to MPT. And with that, we are seeing an increase in ROS. And we have other data that I didn't present to you today, but showing that there's an increase in caspase-3 proteolytic activity on actin, and we're seeing an increase in actin cleavage. And we expect that these are the two major pathways involved in mediating atrophy in general, probably, but certainly in the context of aging, our data is marshaling around that. So with that, I'd like to thank everybody for their attention, thank all the collaborators involved and the trainees in this work, and our funding. Thank you.
Thank you.
So Dr. Pond is co-chairing the session. Maybe would Amber like to lead the discussion?
No, go ahead, Dr. Coletti. You're more than welcome.
Okay, well, anyhow, it's the same job, basically. Are there any questions for Russell?
I see a couple from Carlo Reggiani and Paolo Bernardi are showing their hands on my screen. So Carlo,
yes, no, I had not the. Okay, so
there are 2 questions. Minimum 2 questions.
Yes, I must enjoy very much the issue about apoptosis and atrophy in aging muscle fibers, but I just want to bring the attention to one detail. I mean, atrophy, I understand there is a somewhat understandable mechanism. About apoptosis in itself, in a mononuclear cell, apoptosis means the end for that cell. In a multinuclear cell, like muscle fibers, can be a way to reduce the number of nuclei, and then through this mechanism alter the relation between the nuclear domain and the function protein synthesis, so which is related to the precise size of the nuclear domain.
Thank you. So I agree. The issue that we have with that is that there's evidence that in the context of numerous muscle atrophy conditions, myonuclei themselves don't seem to be lost, even though nuclei in general are being lost. And it's been proposed that probably these are stromal nuclei or endothelial cell nuclei that are actually being lost in the context of atrophy. So it doesn't appear that myonuclear loss is necessary for atrophy of muscle. And this has really been the genesis of our interest in looking at some of the aspects of mitochondrial permeability transition that don't really involve cell death per se. and that maybe activate mechanisms that are more relevant to atrophy in muscle, at least based on what we understand to date.
So that's just, it's not necessary, but it's possible, a reduction of the number of nuclei.
Agreed. Yep.
Thank you.
Thank you.
But sorry, I didn't get it. Did you actually count the nuclei per fiber length or whatever, or not?
We haven't done that ourselves, but other groups have looked at myonuclei in the context of different atrophy conditions. It is on our to-do list to look to see whether or not we're getting a nuclear loss with either the chemical-induced MPT or the disuse model that we're looking at.
Okay, got it. Then maybe Paolo Bernardi had a question.
Yeah, very nice, Russ. It's more of a comment, actually, and I want to stress the importance of one experiment you did. Cyclosporin can be criticized because you can't tell apart the effects on calcineurin, which are so important for muscle regeneration and fiber type switch. But I want to stress that the triazole that you used is not acting through cyclophilin. So the criticism does not apply. So it also validates all your data on cyclosporin, I think. And that's important to stress, that this is really a pore dependent effect. And it's very encouraging that you can see an effect in atrophy. So I liked it very much.